It's a pleasure to be here and participate in the 2022 Harvard Anesthesia Update and to share the stage with Dr. Spies and Dr. Irwin. I'm going to talk about rethinking general anesthesia, and I want to focus on improvements which I think we can make in the near future. And these are the funding sources that I've had the good fortune to have: NIH, for my Department of Anesthesia at Mass General Hospital, my institutes at MIT, and also I'm disclosing that I have financial interest in a company, Pascal, and also Massimo has licensed IP for some of the work that we've done for Mass General Hospital. So this is the outline I'm going to follow. I'm going to talk about unconsciousness maintained by propofol. I'll talk about anesthetic class, mechanism, and patient age. Then I'll say something about multimodal general anesthesia. Then I want to say a bit about closed-loop control of anesthetic state and reanimation. So what is general anesthesia? It's a drug-induced reversible state comprised of antinociception, in parentheses analgesia, unconsciousness, amnesia, akinesia, and physiologic stability. And the key part is this is a reversible state, and we use, as we know, combinations of drugs to achieve this state. It's often said that how general anesthesia works is a mystery, and nothing could be further from the truth. Indeed, one of the mechanisms which we've sort of come to appreciate over the last several years through our research is that the anesthetic drugs create oscillations which impair the ability of the various parts of the brain to communicate. I'm just going to show you a little bit of that now with propofol. So these are the states of propofol-induced unconsciousness. In the awake state, as you see here, you have oscillations that are about gamma oscillations that are about 5 microvolts, and these are high frequency, low amplitude oscillations. And as we move to deeper states of anesthesia, the oscillations become larger and lower in frequency. The amplitude of the oscillation becomes larger and frequency drops. So the anesthetic states are down here from D, E, F, and G. And in fact, if these 5 microvolts, then these oscillations down here are on the order of 20 to 50 microvolts. And in kids, as we'll see in just a bit, they're even larger, maybe on the order of 1,000 microvolts. But these are the states where we maintain patients so that they're unconscious under anesthesia. They don't need to be as far down as burst suppression or isoelectric state, but usually just in the state where you see just a slow oscillation, the alpha oscillation, is sufficient. So what can we say about the alpha oscillations that we see in the case of propofol? Well, our research, both experimental and modeling work, suggests that it's a coherent oscillation that's going back between the thalamus and the cortex. So propofol acts at all inhibitory interneurons throughout the cortex, brainstem, and thalamus, and also as well as in the spinal cord. So this is something of a diagram we could write down even before we've done any experiments. You go to the operating room, you see a picture like this. Well, this is a young woman who's being maintained on a propofol infusion. She got a bolus here to induce anesthesia, and then she has a 10 hertz oscillation here, then slow delta oscillations here. You maintain the infusion, and you turn it off a few minutes later. Let's say over here at about 33 or 32 minutes, she wakes up. And if we look at the whole head and the oscillations across the whole head, we'll see that these alpha oscillations concentrate in the front, and they're quite coherent. And this led us to the idea that this is most likely an oscillation going back between the thalamus and the cortex. That's what our modeling work suggested. Now we verified that through experimental work in non-human primates and rodents as well, showing that there's an oscillation going back between the thalamus and the cortex. The key idea is to understand why this would make you unconscious. The cortex and thalamus being 2 important brain centers for maintaining cognitive processing. If we tie them up in this way, you can easily appreciate why we would become unconscious. However, there's another important feature to propofol-induced unconsciousness, and that's the slow delta oscillations. And these are recordings from patients that have grids and electrodes implanted for epilepsy surgery. And they stay in the hospital for 5 to 7 days, and they come back to the operating room to have the grids and strips removed. At that point, you can do an experiment with them, which is an experiment of nature, because they have to have anesthesia to have the grids removed. And so if you just record the local field potentials I'm showing you here and the spiking activity, pardon me, you can actually see what human neurons do in response to propofol. And that's what we're seeing here. So the colors correspond to the colors on the cortex here, and sort of over the medial temporal lobe. And you can see this is a neuron which has been recorded next to the red electrode. And here what you see is the spiking activity, which is quite desynchronous, as we'd expect for someone during conscious state. And then look what happens when the large slow oscillations come on. you see that the neuron spikes roughly now every 2 seconds or so. So you have this about a half hertz oscillation about a 2 hertz oscillation that you basically see here. So the neuron can't spike where it wants to. It only spikes like now. So this is also quite incompatible with being conscious. And just to show you this in another representation, these are from recent experiments which we've been doing with Earl Miller at MIT, along with his postdoc Andre Bastos and our PhD student Jacob Donahue. This is from Jake's PhD thesis. And so this is the animal, a non-human primate under propofol, or is about to receive propofol, recording from 4 brain areas. So this is 8A, so that's the frontal eye fields, the prefrontal cortex, the posterior parietal cortex, and the superior temporal gyrus, so the auditory areas. So these are kind of 2 frontal regions. This is 2 regions which are sort of more posterior. And so this is the animal awake. You see these nice sort of low amplitude, high frequency oscillations. And the neurons are spiking pretty much wherever they want. To give you some quantitative feel for this, the spike rate here is about 10 spikes per second for these neurons. Now look what happens when the large slow oscillations come on. So these are the same brain areas as you see on the left. Now the animal's unconscious. Large slow oscillations, and look at the spiking. The spiking becomes very sparse and actually kind of coordinated during those sort of upstates across the entire cortex. And what you see here is you have these long periods of down states, like right here. In addition to that, when a neuron is spiking, it's spiking at a much lower rate. So the rate now, over here, it's 10 spikes per second. Over here, it's a half to 1 spike per second. So there's a dramatic decrease in spiking propensity. So spiking activity is necessary in order to communicate so we can transmit information. You can see why the slow oscillation would be a perfect marker of someone being unconscious under propofol. So let's talk a little bit more about how this extends to anesthetic class. So we could look at the EEG here in the time domain, but what we found is it's much more informative to look at it in the frequency domain like this, so you can actually see the spectral properties of the oscillations. And you can see the drugs in the same class have very similar spectral signatures. So the ethers are also GABAergic primarily. They have other mechanisms as well. They have the alpha oscillation, slow delta oscillation. They also have this third oscillation when you go above 1 Mach, which is around 4 to 8 hertz, which is a theta oscillation. The mechanism of that we don't fully understand. It's something we're investigating. And this is low-dose ketamine, which has an oscillation in the 20 to 30 hertz range, sort of like a high beta, low gamma oscillation. And during these states, patients will typically hallucinate. You can see here with dexmedetomidine and just sort of the large slow oscillation. So by using the spectrogram, you can actually track the oscillatory patterns, in a much more informed way, along with the time domain information. And using the fact that every anesthetic has a different signature, you can use this as a principal way to guide drug administration and infer the level of unconsciousness. And we can relate the oscillations, just as we did for propofol, we can relate them to the mechanisms through which the drugs are working in our experiments, so we can have a principal way for understanding the patterns that we're seeing on the EEG. It's not just simply pattern recognition. And then another important point to appreciate is that the spectral properties of the oscillations change very, very dramatically with respect to age. So this is kind of the category we were looking at in just a second ago. This is a 30-year-old gentleman here who has, see the alpha oscillations and slow delta oscillations here. Then here is someone over here who's 57. They're still there. They're a little bit weaker, but they're still quite visible. And then when you move over to this person who's 81 years of age, you can see that they're much, much weaker. The alpha oscillation is rather faint. Moreover, it's important to notice that the cognitive rhythms, which are up here, have dropped in amplitude dramatically. And so this is this is what we typically see, that as patients get to be older, the amplitude of the oscillations become, much, much, much decreased. And this gentleman here is 56 years old. These two guys are roughly the same age. His EEG looks like hers. His EEG looks like his. We age different physically. It makes sense that our brains would age differently as well. And this is, again, why it's important to monitor, because the understanding the particular brain state that the person, what the person really, looks like, we can't just presume that the neurophysiologic response to anesthesia is going to be the same just because two people roughly in the same age cohort. It can be quite different. And you can see here the kids, the 3-year-old and the 14-year-old, They have basically the same patterns as the older patients, but the bands of the alpha, what was the alpha oscillation in the older patients is much broader, and the band of the slow delta oscillation is much broader as well. And this pattern sort of declines through time, with the peak pattern being up around 6 to 8 years of age. So we can use these in the pediatric population as well, but we have to appreciate these age differences across the entire age range so we can properly interpret the EEG and make clinical decisions. And then three other cases which I think are important to appreciate. In very young patients on the order of between 0 to 3 months of age, you won't see any alpha oscillations at all. You'll see just slow delta oscillations. The alpha oscillations don't form until around 4 months of age. This is work that we did with Laura Cornelius Center for Boston Children's Hospital. And what we think is occurring there is that during the 0 to 3 month of age span, the thalamocortical connections haven't formed sufficiently so that you can generate the alpha oscillations. And then another case which is important to see, which I mentioned in the previous slide, is that in older patients you have just sort of slow oscillations under propofol as well as sevoflurane. So this is a very typical EEG pattern of an older patient, just like the 81-year-old lady I was showing you a second ago, just slow oscillations, slow delta oscillations with no alpha oscillations whatsoever. And this reflects probably more brain aging than anything else. We don't have to invoke Alzheimer's or dementia or what have you. But another important case to appreciate is patients that have overwhelming sepsis, like our ICU patients, for example, will have just slow oscillations as well. And we saw a lot of this during the height of the COVID pandemic. This was the sorts of patterns that we saw in the EEG. And so these are important to appreciate, because you'd expect this gentleman here, he's 34 years of age, to have just sort of nice, bright alpha oscillations and slow delta oscillations, just like the patient that I showed you in the previous slide. But such is not the case. And we have here our website where you can download information, or we have educational materials to help teach these ideas on how to use the EEG to track brain states under anesthesia. I'm going to say a few words about some work I've been doing with my colleague Manus Naranjo from Merida in the Yucatan Peninsula in Mexico on multimodal general anesthesia. So this is a paper we published back in 2018. And one of the things that we're looking at is trying to come up with more principled ways to sort of think about drug dosing. And what we came up with is a paradigm which we've called multimodal general anesthesia. Other people have used this term as well, but what we tried to do is come up with a neurophysiologically based approach to selecting drugs. And we figured that the first thing you should try to do when you think about giving anesthesia is think about controlling nociception and picking more than one drug to target the nociceptive pathways. If you do that, then what happens is those drugs will decrease arousal. And what that means is you can adjust the hypnotic dose, the dose of propofol, or the dose of sevoflurane that you're using downward. You can achieve the same level of unconsciousness with less of those drugs. To do this, you have to be able to monitor the level of arousal, monitoring anti-nociception. So for arousal, what we're going to do is we're going to watch heart rate and blood pressure. We're going to monitor the EEG. And for anti-nociception, we're going to monitor heart rate and blood pressure, just the way we do all the time clinically. And ideally, we should maintain a multimodal strategy once we get into the PACU. So let me just go through one case to illustrate this idea. This is a woman I took care of a few years ago. She's over 89 years of age. You can't say the age of a patient who's over 89 years of age because of HIPAA. She has diabetes, hypertension. She's going to have a laparoscopic right colectomy. She's about 5 feet tall. She weighs about 57 kilograms. I used three different drugs which attack different parts of the nociceptive system in order to control her nociception during the surgery. So dexmedetomidine, alpha-2 agent, remifentanil, synthetic opioid, and lidocaine. And what we're able to do is, once the case was underway, after about 45 minutes or so, or about an hour, we're able to control her unconsciousness with just 30 micrograms per kilogram per minute of propofol. What would appear to be like a homeopathic dose. We used rocuronium for muscle relaxation. She had oral Tylenol and some IV hydromorphone postoperatively. So this is just to show you what her EEG looked like. You can see she just has primarily slow oscillations. And as we said, you can just see in the spectrogram that just the slow oscillations predominating here. She doesn't have any alpha oscillations, whatsoever. Not surprising, because she's over 89 years of age. And during the case, her vital signs were quite stable. Well, this is a summary of how we think about this particular paradigm. So the idea is that you pick your drugs that you're using to control nociception. So let's say, picking from this list here. Then what happens is those drugs have a very important secondary effect, which is they also decrease arousal. So they have this effect over here, which means that the propofol that you're using, or the sevoflurane that you're using to maintain unconsciousness, you don't have to administer as much. And one of the key things that we see clinically in this is that the patients wake up in a much more crisply and in a much more sort of timed, predictable manner. We're testing this out now in a clinical trial at the Beth Israel Hospital in Boston with my colleague Bala Subramanian, who's testing out cardiac anesthesia. I just want to say something about closed loop anesthesia and also reanimation. So this is a project that I've been working on with my colleague Earl Miller at MIT, and again, our graduate student Jacob Donahue, one of my postdocs, Chuck Robardi. So we wanted to build a system so that you could use the computer to control the administration of the anesthesia. And we've done this before in rodent models, but the FDA made it clear that in order to test a system like this in a human, we had to do testing in larger animals. So we set up a non-human primate model. And my colleague Earl Miller is an expert in non-human primate neurophysiology. So he was the ideal person to collaborate with. So we're going to set a target level of unconsciousness that we want to achieve, all right? And I'll say exactly what that is. This is called a marker of unconsciousness. That's what MOU stands for. And then what we do is from the EEG, the local field potential in this case, we estimate what that target is, what the actual target is. So the controller computes the difference between the target, and the intended level, and then changes the infusion rate of the syringe pump accordingly, which is going into the animal, just administering propofol. So the infusion is continuous, and the update of the calculation occurs every 20 seconds or so. So it captures the local field potentials, which would be the EEG in a human, and then recalculates what the marker is, and then again repeats the comparison and cycles through like this. And just to give you some idea of what we're going to be looking at, remember I showed you just a second ago for the non-human primates that when the animal became quite unconscious, the spike rates got to be very, very low. And it turns out that if you track the power, this is just an empirical observation, track the power in the 20 to 30 hertz range of the EEG, of the local field potentials, it tracks the spike rates. So dropping spike rates, as I was saying, is a very robust marker indicator that the animal is going to be unconscious. And this is something which we could observe. And the local field potential would track in a very similar way to the EEG. So we're going to use this as our marker of unconsciousness, all right? You can see you give a propofol infusion, you keep it running constantly, and you see how the power just drops. So this is what we're going to use as our marker of unconsciousness. So let me just show you this in a little video here. So this is a target. We've normalized this so it should be at 1, and we're going to raise it up above 1 and below 1. Below 1 meaning taking you into the animal into a deeper state of unconsciousness. Above 1 taking to a less deep state. And then we're going to start off here with the infusion rate. It's going to be constant for about 30, 35 minutes or so. Then we're going to do some changes and let the controller take over. And this is the spectrogram down here at the bottom. So I'll just play the video and talk you through it. So no control yet. You can see the infusion rate is constant, about 0.4 cc's per minute. And the markers sort of wandering all over the place. Now in just a second right here, the controller takes over, comes on, and locks the marker into 1. And it's holding it there, really nicely. Now watch what happens as we decide to take the marker to a little bit deeper. It increases the dose and moves down, so it jumps down to a lower level. It goes down to 0.9. And then from here, we're going to take it up to a higher level, which means a level of less unconsciousness. So it pops up, and it's able to track the marker really well. So what we're seeing here, and one thing I just want to point out here, what the marker did here in order to you can see, look what happens. This is a constant rate. At a constant rate, what you notice is the the animal just gets deeper and deeper. And this is what's happening to our patients under anesthesia. And what the controller did was actually turned off for essentially 3 minutes or so in order to let the marker come back up to the level where it needed to be. And did something similar over here. When it needed to come up to an even lighter level, it actually turned off for 8 minutes so that the actual target could be acquired. And so this is a sort of behavior that's occurring in the operating room when we're taking care of our patients. We're running constant infusions. More often than not, they're probably just getting deeper, deeper, and deeper. So this shows you the benefit of having a control system to monitor the activity of the EEG and use that to control the drug dosing. So one final idea, reanimation. This is the work that my colleague Ken Salt at Mass General Hospital is doing for a number of years, working on ways to turn the brain back on after anesthesia. And he has a rodent here that has an IV receiving a propofol infusion in its tail vein. He has an electrode implanted in the ventral tegmental area, arousal center, dopaminergic arousal center in the top of in the midbrain, the top of the brainstem. And what he's going to do is he's going to turn the microstimulation on here, not turn the propofol off, just turn the microstimulation on. The stimulation is off, he's going to turn it on. And you'll see what happens here. He turns the stimulation on, the animal comes to, starts breathing like crazy. And remember, the propofol is not off. But he turns now, and he runs away. So what Ken is doing is this. So here's this pathway I was talking about. So he has an electrode implanted here in the ventral tegmental area. So this is the mesocortical pathway. Meso meaning midbrain, going up to the cortex. It goes through the limbic system, the hippocampus, amygdala, the nucleus accumbens. And this is a dopaminergic pathway. So when you get Ritalin, for example, it actually acts in this pathway here to help release dopamine. This is one of the reasons Ritalin works in ADHD, or people take Ritalin, or self-administer Ritalin. They shouldn't do this to help improve their cognitive performance. And it's acting in this pathway here. And what Ken did in this case was directly stimulate this electrically. He's also done it optogenetically and specifically stimulating D1 neurons, showing that you can get the same effect of this huge arousal effect in doing that. And in other words, one of the reasons this is such a good pathway to use for that purpose is that it's important for cognition. So if one enhance cognition after surgery, this would be an important pathway to be able to activate. So this is something which he's been working on for a while. And more recently, he just showed that you could do the same thing with D-amphetamine and inversing dexmedetomidine-induced unconsciousness. So some drug in this armamentarium should work as a way to help turn the brain back on and help improve or reduce the incidence of cognitive dysfunction following general anesthesia. So what I've just gone over is a little bit of quickly the mechanism of action of anesthetics, which is primarily, as I said, through creating oscillations which modulate in a dramatic way how the neurons spike, particularly propofol, as we saw there. And then this offers up a very principled way by using these oscillations which are readily visible in EEG and change systematically with anesthetic, with patient age, and also patient state of health. And they can be used as a way to design a control system, as we showed you with the research we've been doing with Earl Miller and the non-human primates. Multimodal general anesthesia as a way to control nociception by having a principled way in which you pick the drugs to control nociception, and the effect that they take advantage of the effect they also have on arousal, so that you can reduce the dose of your hypnotic agent. And then finally, reanimation, turning the brain back on after anesthesia may be a very productive way of decreasing postoperative cognitive dysfunction, and as well as speeding recovery. So I'll stop there. Thank you very much.